I'm here presenting the work comparison of deep learning embeddings of protein sequences for predicting protein-protein interactions. This work is based on the analysis of deep learning embeddings, which are numerical representation of protein sequences, which should contain evolutionary and chemophysical properties. And this is the example of UniRep, SecVec, and ESM1B. For example, SecVec is based on ELMO, which has been developed for natural language process purposes, and it has been trained on very powerful computers. And our question is, can these embeddings be used for protein-protein interactions task? The quick answer is yes, and they can even be used for other kind of prediction task where the proteins are involved. Our dataset is based on more than 5,000 protein-protein interaction that have been retrieved from the DIP database and more than 2,000 known protein-protein interaction retrieved from the Negatome database. And why we use Negatome is because we wanted to have as negative dataset experimentally validated known protein-protein interaction, because we consider other approaches. For example, when non-protein-protein interaction are artificially created based on different subcellular compartments, we think these methods are less accurate. For example, we may miss interaction between membrane proteins of different organelles. We decided to test these embeddings using a step-forward feature selection approach, where, as you can see on the right side of this presentation, the best feature is selected, and as an iterative process, every feature that has been not selected in the first step is combined with the best one from the first step, and so on and so forth until the performance worsen. When we don't have an improvement of performances, the process halts, and the last best performing one is choose. The last combination, best performing combination, is choose. For example, here we see that coupling F5 and F4, we could obtain better performances, but adding an additional feature is not the case anymore. So we need to stop and keep the F5, F4 couple to proceed with. Using this approach, we evaluated 3 different deep learning based embeddings, as I mentioned before, the 3 I mentioned before, and we performed a 5-fold double cross-validation with a random forest classifier to evaluate performances. The double 5-fold cross-validation works in a way that there are 2 different loops, an internal loop and an external loop, where in the internal loop we have the hyperparameters optimization, and the model obtained in this internal loop is then tested against the external test loop that we left out. We saw that the ESMB1 was the best performing embedding here, and we even saw that coupling it with SecVec we could obtain even better performances, meaning that the hybrid combination of these 2 embeddings was better than one embedding alone. But we obtained a very huge embedding in this way, coupling, just concatenating 2 of them. We thought, what could we do? We decided to perform a recursive feature elimination, and that was applied to the best performing hybrid, to optimize the number of features contained in the embedding. And with that, we obtained a very huge reduction, starting from more than 4,000 features, ending up with 1,808 features. And we can see in the second row of this table that the performances are even slightly better, just having this reduced hybrid embedding. So what can we say? We can say that pre-trained deep learning models can actually be used for the task prediction of protein-protein interaction. And this opened the possibility to further analysis. For example, we will test additional deep learning embeddings. We will also create a negative data set based on the previously discussed subcellular localization approach, and we will compare it and also analyze the differences among the two negative data sets. We will also combine multiple deep learning based embedding and test different classifiers, not just the random forest that we analyzed, but we will use also different classifiers such as logistic regression and other machine learning classifiers. Also, we will add every time this recursive feature elimination step to optimize the size of the final embedding, and we will also compare the final predictor against the state-of-the-art methods. We will also aim to train the predictor on protein-protein interaction from other organisms, and even suggest candidates that we could eventually experimentally validate it.